Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're starting a new unit today. This unit is known as the Central Dogma of Biology, and it has to do with this crazy picture on the board up here. Now, this picture might not make any sense to you, but as this unit kind of progresses and as we look at this in more detail, you're going to know all the crazy steps that are happening here. What I wanted to do first is just introduce what does this term central dogma mean, and then we'll kind of go ahead and get started with the lesson, okay? So thanks so much for making it today. Go ahead, give me your attention up on the board up here, and we'll get the ball rolling. So what does this term central dogma mean? Okay, well, the term dogma, that's like something that is true that has been established and is very hard to refute. Okay, so and then a central dogma is a unifying idea. Okay, so what we're talking about over the next couple weeks, this is known as the central dogma of biology. It's something that unifies all of the studies of biology. It links all of that stuff together, and it's been proven and double-checked and triple-checked, and it's very, very hard to refute. Basically, what this means is that everything in a biology class does not make sense if this is not true, okay? It is kind of a complicated process. We're talking about how DNA replicates and how we make proteins using your DNA. There's a lot of steps and some complicated words that go along with this unit, but just follow along with me and I'm going to make it as clear as I possibly can, okay? And what's really funny about this central dogma is that it can really be wrapped up in just 3 simple words. So what is the central dogma of biology? It's basically that genetic information flows in one direction, and that direction is down here in the bottom of the screen. Genetic information flows from DNA to RNA to protein, okay? And those just 3 little words with the arrows in between them kind of link all of biology together. And I'll kind of explain how that happens as this lesson progresses. But what we're really talking about today is DNA and the structure of DNA. So what does it look like? How is it held together? What's the purpose of DNA? That's really our goal today. And as we go forward, we'll figure out how it relates to all of biology. Okay, so let's go ahead and get the ball rolling on this and see what's going on. The first thing I wanted to talk about is the discovery of DNA. This was a huge deal, okay? This was happening in the 1950s and 1960s, so, not even 100 years ago. I know that seems like a long time to us, but some of our parents or grandparents might have been around when all of this discovery was being made. So what's going on is in the 1950s, there's 3 teams that are working really, really hard to try to figure out what the structure of DNA is. At this time, we knew what DNA was, but we really didn't know how it held all of the genetic material. Okay, these 3 teams are engaged in a scientific battle of wits to try to figure it out. Okay, the first bullet point, the first team, this is a pair of scientists at King's College. Their names were Rosalind Franklin and Maurice Wilkins. And their technique to figuring out what DNA looked like was just to take a picture of it. It was a very complex way to take pictures. It's called X-ray crystallography. You use X-rays to take a picture of small molecules, and they were some of the best in the world at doing it. Okay, another team that were working together around this same area, these were the 2 that were actually given credited with finding the structure of DNA, and that's the team that was in Cambridge. Their names were James Watson and Francis Crick. Okay, they're the ones who, they won a Nobel Peace Prize for discovering the structure of DNA, and they're the ones that are really given credit for finding the structure of DNA. So you really want to remember their names, Watson and Crick. I'm sure they'll pop up on our test or the state test at the end of the year. Okay, another person who didn't play as big of a role but was still contributing to the structure of DNA was this individual in America. His name is Linus Pauling. And at the time, he was really the front runner. People thought that he was going to be the guy to figure it out because he already had a Nobel Peace Prize and he was a very well-renowned scientist and he studied proteins. And so when all of this is going on, when they're trying to figure out what is the genetic material, Pauling was really studying proteins more instead of DNA. unfortunately, that was not really the correct assumption, but he did contribute a little bit throughout this whole process, okay? And then in 1962, 3 of these individuals are given the Nobel Peace Prize: James Watson, Francis Crick, and our individual up here, Maurice Wilkins. The 3 of them split the Nobel Peace Prize in 1962, so those are the 3 that really are given credit for discovering DNA. Unfortunately, when this Nobel Peace Prize was given, Rosalind Franklin had already passed away. At the time, they didn't know that X-ray could cause certain types of cancer, and so she's taken all these pictures of DNA and helping people figure out what it is, but at the same time, taking all those pictures with X-ray kind of damaged her body and led to her death. And so although she played a huge role in discovering the structure of DNA, she was never really given the credit that she deserved, because she had passed away, and you can only split a Nobel Peace Prize 3 ways. And so she was never really given the credit that she deserved, in my opinion. Okay, I just kind of wanted to show you a picture. Here are the scientists that are playing this dramatic play out. They're researching and trying to figure out what the structure of DNA is. In the top left corner, those 2 pictures, that's James Watson and Francis Crick. They're studying DNA in Cambridge. In the top right corner, that's the American. His name is Linus Pauling. He's mostly studying proteins. He did not really figure out the structure of DNA. And then at the bottom, this is Rosalind Franklin and Maurice Wilkins, and you can see Maurice Wilkins is actually in front of that complex. It's not really a camera, it's called an X-ray crystallography machine, but that's what they were using to take these pictures of DNA. And so all 5 of these people and many other scientists working together really changed our understanding of biology once we figured out how DNA was structured and how it passed on the genetic material. Okay, here were some other contributions, and you'll hear his name a lot. So this guy named Chargaff, he figured something else out that really opened the doorway for those other scientists to kind of start putting the puzzle pieces together. What he did is he looked at the DNA of a lot of different organisms. So he would take the DNA from all these different animals and different plants, and he was studying them. And what he found out is that in DNA, certain nitrogen bases usually appeared in the same amount. And what he figured out is that the amount of guanine, which is one type of nitrogen base, was very close to the amount of cytosine, which is another type of nitrogen base. He also figured out that these 2 bases, adenine and thymine, usually occurred in about the same amount as well. Now, this ended up being a huge deal, and it really helped Francis and Watson Crick. It really helped them figure out the structure of DNA, but he didn't know what this meant at the time. But it ended up playing a very significant role in their understanding. Okay, so let's talk about deoxyribonucleic acid a little bit. That's the long name for DNA. So DNA stands for deoxyribonucleic acid. Okay, DNA is found in all organisms, plants, animals, protists. Every organism has DNA in it. And for scientists, this is a really strong piece of evidence that supports the idea that all living things share a common ancestor. And we call that common ancestor LUCA, which stands for Last Universal Common Ancestor. Way back in time, if you went far enough, there was probably one organism. It might have been the first one to have DNA in its cells, and then that is the common ancestor for all the forms of life that we see today. For scientists, it's just more likely that DNA came around once and stuck around in all the different forms of life than each individual organism using DNA independently of each other. Okay, so this is the fact that all living things have DNA is a really strong piece of evidence that we all share a common ancestor if you were to go back far enough. Just as a reminder, DNA is inherited from your parents. You get half of your DNA from your mom, half of your DNA from your dad, and this is what determines your physical traits and how they're expressed. So this is why you kind of look like your mom and you kind of look like your dad, okay? So let's talk about the structure of DNA a little bit, okay? So remember that DNA is a nucleic acid, and its job is that it stores genetic information, okay? Nucleic acids are made up of a monomer known as nucleotides, and nucleotides are made up of a sugar, a phosphate, and a nitrogen base. In DNA, there are 4 types of nitrogen bases: adenine, guanine, cytosine, and thymine. And then remember that scientist named Chargaff figured out that adenine and thymine usually occurred in about the same amount, and that guanine and cytosine usually occurred in about the same amount. And we'll figure out why that is here in just a moment. Okay, so some more information about DNA and how it's structured. Okay, DNA is a double helix. That's the structure of DNA, is a double helix. Well, what does a double helix look like? Imagine a ladder and that it's twisted. Okay, that's kind of what DNA looks like. It looks like a twisted ladder. As far as the structure of DNA, the sides of that ladder, okay, is made up of sugar and phosphate. And so we say that DNA has a sugar and phosphate backbone, okay? The rungs of the ladder are the nitrogen bases. So they're the things in the middle, okay, are the nitrogen bases. Nitrogen bases pair up in specific arrangement, which are known as complementary base pairs. So adenine pairs with thymine and guanine pairs with cytosine. This explains why Chargaff found that A and T usually appeared in the same amount and guanine and cytosine usually appeared in the same amount. He didn't know this at the time, but this helped Watson and Crick figure this out later on, that adenine and thymine usually pair together and guanine and cytosine pair together. And that's why you find them in equal amounts. Okay, so let's take a look at this picture and let me kind of run you through everything that's going on. Okay, so this is a DNA molecule. It's not twisted up like it normally is. When I say it's twisted up, when I say it looks like a double helix, this is what I'm talking about. Okay, but this is kind of a close-up of it over here. So a couple things that you need to know. DNA stands for deoxyribonucleic acid. Nucleic acids are made up of nucleotides. Let me point out a nucleotide for you. This would be a nucleotide, this structure right here. This little part with the P, and then this is the deoxyribose sugar, okay? And then this is a nitrogen base, okay? The sides of the ladder are made up of sugar and phosphate. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down, okay? The sides of the ladder are sugar and phosphate. The rungs of the ladder, the parts in the middle, are those nitrogen bases. And they pair up in a very specific way. You can see up here that the green ones are adenine and the pink ones are thymine. And the greens and the pinks always go together. Adenine always pairs with thymine. At the bottom, the blue one down here is guanine, and the red one is cytosine, and they always pair together, okay? So the guanine and cytosine always go together. You will need to remember which nitrogen bases go together, so I'll give you a couple of mnemonic devices on how to remember that. Some people just say apple tree and garage cat. So you can think of that if you want to, or car garage even. I just remember the word at, A-T, like I'll meet you at school. To me, that makes it really easy to remember that A and T's go together and C's and G's go together. Whatever works for you, fine. You can think of apple tree and car garage, or you can just think of the word at, okay? A and T's always go together, C's and G's always go together, okay? So let's talk about those nitrogen bases in a little bit more detail, okay? Some of the nitrogen bases have 1 ring, and some of the nitrogen bases have 2 rings. And depending on if it has 1 or 2 rings, they get different names, okay? So purines are the nitrogen bases that have 2 rings in them. And examples of those are the adenine and the guanine, okay? For the pyrimidines, if it has 1 ring as the nitrogen base, we call those pyrimidines, and those are thymines and cytosines. And so we always get a purine and a pyrimidine to pair together because the adenine always pairs with the thymine and the guanine always pairs with the cytosine, okay? And again, A and T go together and C and G go together. So if I go back to this picture, you'll see that there's always 3 rings in the middle, right? 1, 2, 3, 1, 2, 3. That keeps it even. We don't want more rings on one rung than the other. The rungs would be different lengths and it wouldn't work. It wouldn't keep its shape very well. Okay? Great. Okay, so the last thing, let's talk about what's holding this molecule together. DNA, it's very long. The structure of DNA is very, very important. And so holding it in its correct shape is crucial to its job. Okay? In between the nitrogen bases, the rungs of the ladder, the thing that holds each side of the molecule together are these weak hydrogen bonds, okay? And we actually want these bonds to be really weak because sometimes we have to unzip the DNA molecule. And having weak bonds in between the nitrogen bases makes it really easy for the DNA molecule to unzip. Okay? Along the sides of the DNA molecule, we have these really strong bonds. And these strong bonds are known as phosphodiester bonds. Those are very, very strong. And we want the backbone of the molecule to stay intact. So we have weak bonds in between the nitrogen bases and really strong bonds in between the sugar and the phosphate. And that helps hold it together where it needs to be held together, and it's easy to break apart when we need to break it apart. We'll break DNA apart when we are replicating it or when we start to make proteins. Okay? Y'all, thank you so much for following along. Your teacher has some other work for you to practice this stuff on, so make sure to give that your best effort, and I hope everybody has a great day.